వాడు ఏమంటాడు వాడు ఇప్పుడు అంటే మనం లేట్గా జాయిన్ అయినాం కాబట్టి వాడు డిలీట్ చేసినట్టున్నాడు గురు. ఏది నంబరా? నువ్వు థర్డ్ సెమ్ అనేది నీ ఫస్ట్ సెమ్ నంబర్ అంటే మరి ఆగు.
ఇంగ్లీష్ మీడియం
మరి బుక్ ఏది?
ఇంగ్లీష్ మీడియం, తెలుగు మీడియం.
నువ్వు ఏ సెమ్?
ఫస్ట్ సెమ్ ఇంగ్లీష్ మీడియం.
ఫస్ట్ సెమ్ ఇంగ్లీష్ మీడియమా?
రెండోదా ఫస్ట్?
ఫస్ట్దే. ఫస్ట్ సెమ్ కదా? ఐడి 963 6890 97 16 16. ఒకసారి కొట్టంగానే వస్తుంది కదా.
నెక్స్ట్ పాస్వర్డ్ చెప్తారా?
156698 156698.
అక్కడ పంపించద్దు.
Okay. Good afternoon, everybody. Now we discuss the genetic code and protein synthesis. So here, genetic code protein synthesis, that is the central. The molecular biology starts the here genetic information from the DNA to RNA. Okay. So that is the DNA in the transcription to form the RNA, and that is the translation to protein synthesis are there. So what are the types of the RNAs are here? Different types of the RNAs are present. There are the here, see here. There are the three forms of RNAs are there. That is the ribosomal RNA, transfer RNA, and messenger RNAs. These are the made on the DNA templates. The enzyme RNA polymerase catalyzes the synthesis of RNA. The three forms of RNA, that is rRNA, tRNA, and mRNA, this is distinguished on the basis of their molecular size and function. There are the defined to in the terms of the segmentation constants, which is proportional to the logarithm on the molecular weight here. That is the mRNA means a single strand mRNA molecule forms a small amount of the total RNAs. So in this, it carries the messenger to the ribosomes, hence named as a mRNA. So this RNA is synthesized rapidly to break down the rapidly on the basis composition of mRNA. Similar organism reactions, the thymine is replaced by the uracil. Instead of the thymine, uracil is present. So it is the present in small amount of the cell. Many ribosomes are attached to the mRNA strands called as a polyribosome. So another one is the rRNA, that is the ribosomal RNA, site of the protein synthesis, where the amino acids are assembled in the cell sequences to produce the polypeptide chains. Okay. So, so the ribosomes were first observed by the ribonosine and Brown in the plant cells are there. So the ribosomes are occur freely on the cytoplasm and also attached to the endoplasmic reticulum. That one we call as a rough endoplasmic reticulum. So both the free and membrane bound ribosomes synthesize proteins. These ribosomes attached to the endoplasmic reticulum are active in the protein synthesis. These ribosomes consist of approximately the equal part of the RNA and proteins in eukaryotic cells and catalyze the incorporation of the amino acids into protein in the presence of ATP and magnesium and other some cofactors also there. Okay, so here here that is the cytoplasmic ribosomes of the plant cells are in around 20 nanometer diameter, contain 40% of RNA and 60 proteins are there. This molecular weight is about 4 into 66 times. The ribosomes are composed of two subunits which are unequal in size, that are the small unit lies beside of the large and subunit. A characteristic cleft divide two subunits into unequal regions. The magnesium ions are necessary for their structural and cohesion. So when the magnesium is limited, the ribosomal unit break down into particular and became the inactivate in the protein synthesis. The ribosomes constantly dissociate into two subunits, and the dissociation is essential for the protein synthesis. When a number of ribosomes is sitting together in the RNA strand, it is known as the polysomes or polyribosomes. In the higher plane, the polyribosomes may be major site of the protein synthesis. The presence of ribosomes in which protein synthesized, the RNA is present in ribosomes 3 on the cytoplasm and ribosome attached to the endoplasmic reticulum. The rRNA constitute about the 3/4 of the RNA in the cell. The rRNA molecule are single strand polynucleotides which are unbranched and flexible. There are the coiled folded within the ribosome. The base composition of rRNA shows generally similar from the organism to organism. The next one, tRNA, that is transfer RNA, is known as called as a soluble RNA. So it is designated to tRNA more appropriate became it is involved in the transfer of amino acids from the cytoplasm into the proper position on the ribosome. So the tRNA forms 50% of the total RNA of the cell. The molecules of tRNA are the smallest and contain about 75 to 90 nucleotide bases. Their molecular weight is about 25,000 to 30,000, and these at least one piece of RNA, then 20 amino acids forming proteins. They are synthesized on the DNA and released into cytoplasm. So here the next one is the genetic code. So genetic code means here so genetic code, there are the 20 different kinds of amino acids which are formally constitute the different proteins. The single and double base coding will not cover all the 20 amino acids. A single genetic code involves each nucleotide. The coding for a single amino acid can code only for the protein with the 4 amino acids, while the two bases code for 16 amino acids in the formation of the polypeptide. It was suggested by the Crick, and it has been proved experimentally by the Nerberg and Mackey. So there are the listed different types of the codons are there. So what is the features of the genetic code? Here the code is the degenerated for a particular amino acid more than one codon and used except for the methionine and tryptophan. The code is non-overlapping. So in overlapping code, change in one base could affected more than one code. So that's affected more than one amino acid. Since a basis in mRNA is not used for two different codons, that is non-overlapping. The code is commonly when the triplet codes are amino acid, the second amino acid is automatically coded by the next triplet and no base where the wasted in inverted sequence. So then other character that is the universally the genetic code. So that is the triplet codon for the some amino acid is all living organisms. However, the description was found in mitochondrial genes. So Webel hypothesis, this is the less important for the third position of the genetic code. The first base of the anticodon may be able to pair with any several bases in their position of the codon. This is known as the Webel hypothesis. So So here protein synthesis. Hmm? So this is the protein synthesis. In the process of protein synthesis can be split into two stages. There are the transcription and translocation is there. These are the two steps are there. So in the transcription, the synthesis of RNA complementary to strand of DNA is known as the transcription. So DNA complementary, that is the transcription. So the transcription again divided into three stages are there, that is initiation, elongation, and termination. So what is initiation here? To begin within the process of transcription, the RNA polymerase identifies the transcriptional start site with the help of the promoters. The two key promoter sequences are located approximately 10 nucleotides and 35 nucleotide upstream from the start point. The DNA promoter region is prokaryotes is a starch stretched for about 40 bases pairs adjacent to and including the transcription start point. The essential feature of the promoters are the start point, that is there. Okay. So next one is the so initiation. This is the initiation there. The next one is the elongation. 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 See here. So elongation means the enzyme RNA polymerase catalyzes the synthesis of RNA by using the nucleotide triphosphate, that is the ATP, GDP, CTP, and UTPs also. Though during the elongation, the RNA polymerase binds to about these 30 base pairs of DNA. So at any given time, about 18 base pairs of DNA unwind, and most recently synthesized RNA is still hydrogen bound to DNA, forming a short RNA DNA hybrid. The total length of the growing RNA bound to enzyme and DNA is about 25 nucleotides. So the termination, another one is the termination here. So in the termination of the transcription is requires a termination sequence, that is the triggers end of the transcription. Broadly, the termination of the transcription can be either row or depending on the row dependent process. The row factor is an essential transcription protein in prokaryotes. So that is row factor act as a ATP dependent unwinding enzyme, moving along with the newly forming RNA molecule towards it three hands and unwinding from the DNA template. It is the proceeds. So in the row terminate depending on the row to mRNA release in the form the template. In the row dependent termination, a short complementary G-series sequence and will form a break that will help release the RNA polymerase from the template. The process of transcription is eukaryotes and prokaryotes. Okay. Mhm. There, except for some minor difference in the terms of enzyme, involve the additional protein factor required on unwinding of the DNA and identifying the promoter sequence. So the translation. Next one here, the translation process. So this is the termination, and translation means this one is here. So the translation can split into four stages are there. The formation of the initiation complex. So the translation starts with the amino acetyl RNA carrying the N-amyl methionine pair with the initiation codon AUG located on the 60 the 30 subunit of the ribosome at the P site. The resulting structure is the comprising mRNA and 30th subunit and aminoacylated the tRNA meet to called as the initiation complex. This is the initiation stage of the translation. The initiation in E. coli require the 3 points called as the initiation factors. So that is the IF2 or participation in the attachment of the tRNA and its binding to initiation complex, which is with a molecule of GTP providing energy. The initiation complex bind to the site and leaving the seconding bind AFN. The elongation of the polypeptide chain. The next tRNA molecule recognize the next codon sequence and mRNA itself on the A site of the ribosome. The next tRNA molecule recognize the next codon sequences on mRNA and it attaches to the open A site of the ribosome. This requires another GTP and two elongation factors. The elongation being when the correct amino acid tRNA enters the A site and the pairing takes place with the second codon. Peptide formation and translation. Then both sides of the ribosomes and occupied by the amino acylated tRNA. The next step is the formation of the peptide between the carboxyl group of front and amino group of the second amino acid. This reaction is catalyzed by the peptidal transformation, which breaks to the femto RNA link as a result in peptide attached to the tRNA presence in the A site. Okay, so the translocation now occurs. The ribosomes moves into the mRNA by length of the 3 nucleotides, so that tRNA with the polypeptide in the A site is moved into the P site. The tRNA deplete to ejected from the P site and empty A site is available for the next tRNA, and the elongation cycle is repeated. Each cycle is require hydrolysis of the molecules of the GDP and is controlled by the elongation cofactor, that is the EFG. Each mRNA can be transmitted by the several ribosomes at one time. The peptide bond formation on the ribosome between the N-formal methionine and polyphenylamine. The bond attached to the carboxyl and the first amino acid to its tRNA is transformed to the amino end of the second amino acid. The first tRNA is now attached, whereas the second tRNA has repeated attached. So now the tRNA with the peptide in the P site and A site is again open. In this, the next tRNA moves into the A site carries glutamic acid. So this is here. Okay. So the chain termination, the termination of the protein synthesis occurs when the termination codon, that is UAE, UAG, and UGA enter the A site. The protein is called as the release factor that is involved in the termination of the GTP is hydrolyzed to GDP and inorganic phosphate. When the nonsense codon enter into the A site on the ribosome, it is recognized by the release factor that give and complete the polypeptide. The ribosomes dissociate into 30S and 50S subunits that will enter into the new round of the translation. The polypeptide may be modified by the attachment of the chemical groups the polypeptide chain fold into tertiary structure and begins its functional life in the cell. A nonsense codon is the site for recognizing the both to release factor in this case. So here, um, the AG and recognized by the RFI, the complex then falls apart, release the peptide. In prokaryotes, where no nuclear envelopes exist, the translation begins before the transcription is completed. Antibiotic like tetracycline, puromycin, and chlorophylamine block the protein synthesis. The eukaryotic transcription result in a primary transcription before entering into the cytoplasm. RNA processing takes place by removal of the intervening the sequence, modification of the 5 to 3 ends and caps into the tails respectively. Thus, the mRNA transcript from the DNA is called heterogeneous nuclear mRNA or RNA that will have post-transcriptional modification, unlike the prokaryotic synthesis, where the translocation begins, the transcription is completed. So next one, the proteins. So here the proteins are the organic proteins are the organic compounds are there. See here, this is the see here, this is polypeptide chain. So in animal, plants, and bacteria, they are the large macromolecule consisting of number of polypeptide with the molecular weight ranges from several thousands to several millions are there. The protein contain carbon, hydrogen, oxygen, nitrogen, and frequently sulfur, and rarely the phosphorus and iron is also present. The building blocks of the proteins are the amino acid, and they are grouped into macromolecules called as the polypeptide. The primary sequence of polypeptide chains are composed of a different amount of the 20 the amino acids. The peptides bond which link into the amino acid together is formed by the consideration of the two amino group. The amino acid with the carboxyl group of another. The proteins have localized the hydrophilic and the hydrophobic area and have defined site for the nucleophilic and electrophilic displacement, which import to them the unique to catalytic activity and their molecular affinity. The proteins have four structural level of the organization, which are referred as the primary, secondary, tertiary, and quaternary structures. So what are the primary structure? It is the linear polypeptide chain formed by the linking sequence of amino acid by the peptide bond. And the secondary structure, each peptide chain may form sheet-like structure or adapter, a coil or helical structure. The sheet-like structure is formed by the hydrogen bonds between the oxygen and nitrogen atoms of the different groups of the polypeptide chain. There are the configuration of the collectively known as the B structure, as described by the pulling and used the X-ray diffraction method. The regular arrangements of the polypeptide chain is brought by the hydrogen binding between the groups of the some chain with the result in the twisting, forming an uh, X helix. The tertiary structure of the protein is three-dimensional form. In many cases, irregular or containing both the helical and non-helical region. The tertiary structures are folded into the different configuration by the formation of the hydrogen bond, hydrophilic bonds, and disulfide bind between the H group. The tertiary structure of protein is essential for the biological activity. So, what is the quaternary structure? The quaternary structure is composed of two or more polypeptide subunits which are non-covalently linked. Of the four polypeptide chains, in it is each of these two are composed of two traces are there. So this is the the gene code and synthesis of proteins. So in summary, the central that is the molecular biology leads to the transfer of the transformation and from the DNA to RNA is directly control the protein synthesis. That is the translation. There are three forms of RNAs are there, which is molecular size and function. mRNA is the and rRNA and sRNA. Okay. So the next one is the mutation. So okay. So that is the black fourth unit. That is the black 4. From that black fourth here 10th unit we discuss that is the mutations. So mutations means the mutations are any heritable changes in the base sequence of DNA molecule. Okay. So the most common types of the changes is substitution, addition, and deletion on more or basis. The integrity of DNA is very vital to the cell and survival of the species depend on the upcoming maintaining and nucleotide sequence intact. Though the genes are made up of DNA, a gene is the entire DNA molecule that control is the particular reaction of the cell. Together, all the genes of an organism control in the life activities at each cell division. The DNA molecule of the cell also get and duplicate occurrently. At each cell division, the DNA molecule cell duplication occurrently. Okay. So here the Hugo de Vries was used the term mutation to describe the phenotypic changes which are heritable. So what are the types of the mutations are there? So see here, now the different types of the mutations are present. What is this here? The five types of mutations are there. So types of the gene mutations means here spontaneous and induced mutations also there. Here, so depending upon the types of gene mutation, it will either the protein produced or will affect of DNA resulting structural mutation. The based on the effect of organism, the mutations have been classified into categories are the morphological mutation. This involves changes in morphology, including color, shape, size. That is another one is the lethal mutation. This involves the genotype changes leading to death of individual. Example, albino mutation resulting from the chlorophyll deficiency in plant. Okay, lethal mutations means the genetic genotype changes, and biochemical mutations. The biochemical mutations are identified by the deficiencies, so that are the defect can be overcome by the supplying the nutrition or any other chemical compounds for which is a mutant is deficient. Another one is the resistant mutation. The resistant mutations are identified by either ability to grow in the presence of an antibiotic or pathogen, to which is widely type is suspectable. So here the physical mutations and chemical mutations also there. The physical mutations, visible light and X-ray or gamma rays, beta rays, neutrons, UV rays also there are effects. And another one is the spontaneous mutations. So spontaneous mutations also there. That is the spontaneous mutations in the see here the spontaneous mutations. Watson Crick is suggested the mutation occurs spontaneously during the process of DNA replication. So this is the spontaneous. Then physical mutation, we following the different types of physical mutation is the ionizing radiation, alpha, beta, gamma, or another. So that is the X-ray is high energy wavelength. They are they penetrate the atoms of the molecular and release the electrons. So this is the so chemical mutations are there. That is the chemical mutations means there are the different types of the chemical mutations also there. Here the base analogues the chemicals that are structurally similar to base in DNA but may have different base pairing protein from for properties are there. That is the bromouracil is structurally similar to thymine. So we will incorporate in a growing DNA stand in the place of thymine, but due to the properties is with base pair more frequently with the guanine than thymine. The mutagenic effect mostly due to the incorrect base the pairing with guanine leading to GCAT transcription in guanine cytosine adenine. So base modifiers, the chemical that make the changes to specific base changing its ability to base pairing property, that is the deamination of the cytosine creates an uracil base will the pair with the adenine instead of the guanine. So intercalating agents, the chemical that is insert themselves into DNA helix causing and DNA's replication and transcription problems usually result in deletion. There are the deletion and insertion. So other mutations also there. That is the carcinogenic agents. That is the nickel carbonyl and these also other agents. So gene mutations are point mutations during the so here gene type mutations are there. That is the here point mutations also we call as the frame shift mutations and another one is the the base substitution mutations are there. So the frame shift mutation which result the shift in the reading the frame caused a new few nucleotide creates numerous missense and nonsense codons downstream to the mutational event. And again, this is two types are there, inversion and deletion. So in this insertion, this addition a pair of more nucleotides to the DNA. So deletion, this result in missing the nitrogen base or nucleotides of the DNA. So insertion and deletion. So another one is the base substitution mutation. This result in the substitution one molecule, and another it may be one purine and replaced with another purine. That is the either one pyrimidine or replaced with the another pyrimidine. Okay. So another one is the transitions. In this one purine is replaced with the another purine, that is the adenine thymine base pair is replaced with the cytosine and guanine. So that is the transition. So another one is the transversions. In this, one purine is replaced with the another pyrimidine, vice versa. That is adenine and guanine is replaced with the cytosine and the thymine base. Some chemical agents like ethyl methane sulfate and ethyl ethane sulfonate causes the removal of the purine rings from the DNA. It begins with the ethylation of purine ring and end with the hydrolysis of the glycosidic bond. When the DNA replication takes the place, the DNA polymerase third insert any four base in the new. So gene mutation in hemoglobin, best example for the point mutation consists in the deviation disease in humans and sickle cell anemia. The sickle cell anemia in human due to the base pair of the substitute with the result of abnormal hemoglobin. And hemoglobin S, the hemoglobin S is not efficient enough for carrying the oxygen and individuals suffer from the disease called sickle cell anemia. So the genetic disorder, this is red blood cells are sickle shaped and victim suffering all the symptoms of extreme oxygen shortage, that is weakness and mutation, kidney and heart failure condition. So another one is the chromosomal aberration. So chromosomal aberrations, there are the like gene mutation, there are the here following two types are there, that is the change in the number of chromosomes, change in the structure of chromosome, that is the chromosomal aberrations. So that is the change in number of chromosomes, so that is euploid and polyploid is also there. So euploid, polyploid, in these cells are high plants and animals, there will be two sets of chromosomes. One is derived from the male gamete and another one is from female gamete. The set of the chromosomes to form one gamete is called as the genome. So thus the cells are higher plants are diploid. We may say have two genome. The number of chromosomes for each plant and animal is fixed. So the chromosomal mutation that is involves a set of the chromosome is called euploidy. If the number of set of chromosomes in any plant is more than two, then it is called as a polyploid. Such plants are called as a polyploids. So it is very widely seen in the plant world. That is the essential near half of the plants are generally are polyploids. So many our economically important plants are polyploids all there. That is the banana, pineapple, tobacco. This all are the polyploid plants are there. So So in polyploids, there are two kinds of. Some of the polyploids are raised in the result of duplication of the chromosomes, set of the same species. They are called as autoploids. When the chromosomes assets of two or more species combine to form the polyploid, it is termed as alloploid. Allopolyploid. So polyploids may rise among the diploids due to any of the following reaction. That is, the all egg cell may get fertilized by the more than one sphere. Second one, as a result of the failure of meiosis in spore mother cells, the germinates will be diploid. Fusion of such gametes result in the polyploid. So during the mitosis in a diploid somatic cell, if the chromosomes duplication is divided but there is a failure in cytokinase and tetraploid cell forms, the normal division of such cell give rise to the tetraploid tissue. The flower borne such as plants produce diploid gametes. Fusion of such gametes bring the tetraploids. These triploids are formed when the two gametes, one with the haploid number of chromosomes and another one the diploid number of united. So, but these plants are meiosis higher irregularly sterile in the prevent. However, such plants are can be propagated vegetatively. For example, musa, rose, and chrysanthemum plants. In the induced polyploid condition, here there are some agents applied induce the polyploid by applying some chemicals and through temperature shock and causing the mechanical injuries to tissues. Polyploidy can be induced. The chemicals like colcin and believed to interfere with the spindle formation, cell division, thus leading the polyploidy. The colcin is an alkaloid prevent in the bulbous of the colcin. This is an applied to birds, roots, and plants in every diluted aqueous forms. So even cytokinesis is prevented, and with the duplication of the chromosomes, their number goes on the increase in the tomato plant where the stem strips are cut and disorganized into cell division takes place. The cell callus form most of the cells such as tissues and tetraploids, while the rest of the plant is diploid. This mechanism injury also induced polyploids in the plants like maize. Polyploidy can be induced temperate shocks which are disturb the cytokinesis. So the variation in the large number of genes in polyploids than diploids. Haploids. Next one is the haploids. In all the higher plants and animals, organisms diploid and haploid is confirmed. Only two of gametes, but occasionally haploid plants are seen in nature like gametes. Those are one set of chromosomes. In the angiosperms, haploid plants are the brassica, datura, gossypium, and oryzae, GMAs. In animals, haploids are very rare. That is there. Honeybees, ants, these are the applied cells. So heteroploid or aneuploid. Sometimes some organs may have chromosomes which are favor are more than exact multiply basis number of applied number of chromosomes of the organism. Such an organism is called as aneuploid or heteroploid. The number of chromosomes in such as the plant may be little more, either less in number of diploid number. The heteroploid are seen the plants, animal, human beings. For example, datura, maize, wheat, tomato are example the heteroploid. In the animals, that is the Drosophila, several kinds of heteroploids are seen. Sometimes the gametes are same species may have different haploid chromosome number. The fusion of such gametes result in the heteroploidy. Irregularity in meiosis result in the formation of the gametes with a different chromosome number. This may be due to non dissection and homologous pair during the meiosis, as a result of which both the chromosomes reach on pole only. At the beginning meiosis, there may be failure in the pairing of homologous chromosomes. So the gametes formed may be variation in number, and gametes may be fused and result in the heteroploid condition. Trisomic condition, that is the trisomic condition, they have one extra chromosome, that is the 2n plus 1. One chromosome may be three homologous chromosomes. If there are two extra chromosomes belong to the two different chromosomes are there, that is the trisomic chromosome. Tetrasomic chromosomes means 2n plus 2, means four homologous chromosomes of one set its chromosome, and monosomic chromosome. In these plants, the number of chromosomes will be one short of the diploid number. That is the nullisomic. Another one is the nullisomic. In these plants, one entire homologous pair may be missing, means 2n minus 2. There are the formed self correlation, and another one is the applied in man. So these are 46 chromosomes. Sometimes they are aneuploid. The aneuploid is seen in such individuals. Such a person may be less than 46 chromosomes. That is the aneuploid condition is there. Mm, the another abnormality when the individual have the exact chromosome, that is the from 47 chromosome, they have three sex chromosomes, is additional called as a Klinefelter syndrome. The male have this syndrome have very small testes with the slightly developed breasts. In some individual have 47 chromosome, that is the 21st chromosome have three homologous chromosome. This is known as a trisomic, result in the Brown syndrome, which are individual called as magnolidae. These are the deletion of even single chromosome will have serious sequence of the individual. So changes the structure of the chromosome. The exception, that is the some total characters of organisms is determine the number of sequence genes present in the chromosome in its cells. We have seen the change in the number of chromosomes affected in the character organism, like the changes in the architecture and morphology of the chromosomes also affected in the characters. So what is the changes here? The deletion and deficiency. So sometimes a part of the chromosome may be get broken. This may be happen in the tip of the chromosome or anywhere in the chromosome. The duplication. This is the presence of part of chromosome excess. The normal completion, it is called as a duplication. A broken section of chromosome may get attached to the normal homologous chromosomes. Consequently, some genes are present in more than double those. That is the duplication. And another one, change in the involved arrangement of genes, that is the inversion. This is the breaking the chromosome into 2 points. The broken section, which is rotates around 180 degrees axis. Another one is the translocation. Huh? So So translocation, this is involved the transfer of short part of the chromosome, a set of genes to a non-homologous chromosome. It results in a change in the sequence and the position of the genes, letters to two non-homologous chromosomes. And another one, the cytoplasmic mutations. We have already seen the plasma genome, that is the cytoplasm. It is established that is the organelles known as the chloroplast mitochondria. They have the DNA. So these plasma genes also get mutated and transmitted character accordingly. The variated part of the plants are known as recognized due to the mutations in chloroplast. So that is the next one, frequency of gene mutations. So this frequency of gene mutations also there. What is the significance of mutation? So unboundedly, the mutation is the principal factor in evolution trends. It may mainly due to the mutations that are the result in some genetic variability. Without evolution means not possible. It is also true that very high percentage these mutations are harmful and small percentage there are useful. In this useful, the mutation bring about the changes in the organisms so that can be adapted themselves to environment, can stand competition in the natural selection. So natural selection so that the progeny survive. All the extinct plants and animals are those formed due to the mutations and have come to the successful in the struggle for survival. Thus, natural mutations have played and continue to play the crucial role in the evolution of organisms. The induced mutations are useful to developing the new varieties of plants suitable to different environmental conditions, and many variations are before it is possible on the plant breeders to select those who have to desirable characters for his hybridization. So experimental through which are can raise the superior varieties we can choose. Okay. So thank you.
Hello, sir. Several important questions. Sir,
దా ఉన్నాయి ఇంకా ఏమున్నాయి అమ్మా లాక్ వేసేసి అంతే ఇంకున్నాయా అంతే కదా అయితున్నా అయింది కదా 4